Hi guys, so today we are going to learn about protein synthesis, and I'm sure you have heard of the word proteins before. Most so in when we were talking about macromolecules, and we said that protein is a macromolecule. Synthesis, Synthesis, basically is the making of proteins. So that is what we're going to learn about today, the making of proteins. What is happening in our cells in order to make proteins, and why do we need proteins? So let's list all the concepts that we learned about already. We learned about DNA, and we said that this is our genetic information. Now, the instruction for making proteins are actually held in our DNA. That is why I am bringing in this concept of DNA to protein synthesis. We already learned that inside of our cells, the DNA is held in the nucleus. And we said that they are tiny dots that are called ribosomes, R-I-B-O-S-O. And here is exactly where the proteins are made. So the nucleus contains the instruction for making proteins, and they are actually made in the ribosomes. We also learned that DNA contains other instructions that is not related to protein synthesis, but we're going to only focus on the information that is used to make proteins. We learned about some of the things that are made up of proteins. We said that our hormones, enzymes, and even our hair, nail, skin, they're all made up of proteins. So these proteins are made in the ribosomes. These are concepts that we already know. So let's talk about protein a bit more. Proteins are large molecules. Now these molecules are very, very large, but they're made up of smaller molecules. Now these small molecules are called amino acids. Amino acids. So this is the protein, this is large. Smaller units called amino acids actually make proteins. Now, when these small units come together, they can form small chains, and these chains are called peptide. So if we break a protein, we will most likely get peptides. If we break peptides, we will get amino acids. The Regents like to bring questions such as what are the building block of proteins? So basically that is asking you what makes up proteins, and if you say amino acid, you will get that correct. To start our process of protein synthesis, we're going to look back at our DNA. So step one. Step one is that we have our codes. So we're going to use the codes that are here. The codes are A, T, G, A, C, G, G, A, T, C, A, G. And just like, let's just use this sample. So this is our strand one. And when we talked about replication and we talk about DNA, we said that A pairs with T and C pairs with G. So if we have to make strand 2, and this again is a revision, is we will have A pair with T, T pairs with A, G pairs with C, A pairs with T, C pairs with G, C, C, T, A, G, T, C. So this is strand 2 of DNA. Now, the first step is that this DNA molecule will divide and or it will unwind. So we will take one strand. So our DNA strand that we're going to take, let's take this strand A, T, G, A, C, G, G, A, T, C, A, G. Now this is found inside of our nucleus. This is the information that we will need to make the protein. So think of it like a recipe. This is the recipe that we will need to make our protein. This step is called also transcription. I'm gonna write that here. Transcription. To transcript or to scribe is basically to write. So basically, the DNA is going to unwind itself, and we are going to copy this strand. Now, something called an mRNA is going to be found. Now, the thing is with mRNA is that A pairs with U and C pairs with G. There is no T in RNA. So A will pair with U, T will still pair with A, G pairs with C, A pairs with U, C pairs with G, G pairs with C, G pairs with C, A pairs with U, T pairs with A, C pairs with G, A pairs with U, and G pairs with C. This strand that is made is now called an mRNA, which is a messenger RNA. M means messenger. So this messenger RNA will travel outside of the nucleus, and it will take that code to the ribosomes. So let's review this process. So this is step one. The DNA strand is divided, and one of the strand is copied by the mRNA. This is called transcription. When the mRNA copies the strand, there is no T. Instead of T, there is a U. The mRNA will move outside and take the strand to the ribosomes that is found outside of the nucleus. So there are three types of RNA. There is the messenger RNA, which copies the DNA code and carries the genetic information to the ribosome. That is what we've been talking about. There's another type of RNA. This is called rRNA. And along with protein, this is actually what makes up the ribosome. And finally, there is a tRNA, which is a transfer RNA that will bring the amino acid that is coded by the mRNA. Just to recap, that RNA contains the base uracil or U. DNA has the code T. There is no U in DNA, and there is no T in RNA. RNA is a single molecule, whereas DNA is a double stranded molecule. Now, once the DNA is outside, the tRNA will bring the amino acid. It will bring the amino acid for the code that we have here. So the code is U, A, C, U, G, C, 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 U, A, G, U, C. Now each code is read in triplets. So when the tRNA comes and read, it will read each one. Each code is called a codon. Each three letters called a codon. So the codon is read. So we're going to look at our chart, and we're going to figure out what amino acid it will code for. So UAC, AC is here, UAC. So it will code for the amino acid TRY. Then we're going to take the next three codons, which is UGC, UGC, and UGC. It is here. UGC is coding for the amino acid CYS. The next three is CCU. CCU is code for the amino acid PRO. And AGU codes for the amino acid SER. So as you can see, it's translated, it's read now. So this is the second step called translation. 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 So translation is reading the codons to make the appropriate proteins, and the tRNA brings that amino acid after it reads the message from the mRNA. I know this is a little bit confusing, it's a lot, but we're going to practice some questions and I'm sure you're going to get it correct. Questions. Let's start off with a basic question. The basic question is, where is the information that is stored to make protein? And it's stored in the DNA. Next question is, where are proteins made? They are made in the ribosomes. The next question is, what is the first step in protein synthesis? It is called transcription. And in transcription, the DNA molecule will unwind and you will have a single molecule. After you have a single molecule, you will make an mRNA. Now that means that the code will be read from that single molecule. Now in the mRNA, we have to remember that A pairs with U and C pairs with G. Now this single strand of mRNA, so the mRNA will move outside of the nucleus and it will go to where the ribosomes are. It'll go to where the ribosomes are. Now, I'm going to give you an example. So say, for example, let's work this problem. If they give you a DNA molecule with A, T, 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 G, C, C, G, A, C, G, A, T, A, this is DNA. They ask you for mRNA. All we have to do is figure out the codes. So A pairs with U, T pairs with A, T pairs with A, T pairs with A, C, G pairs with C, C pairs with G, C pairs with G, G pairs with C, A pairs with U, C pairs with G, this is C, this is U, this is A, this is U. Now each one is from a codon. Each three letters, sorry, is form that is formed is called a codon. So we will have to use this chart, and this one they will give you a chart to read it. So UAA, if we go back to our chart, UAA, where is UAA? UAA is a stop codon, so it will form STOP. ACG, ACG will form THR, and so forth. So this is your amino acid.